All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So today we have a very cool in vitro study from Japan. In vitro study from Japan. It is not only about the ADE. I think there are many studies now that discuss the presence of antibody dependent enhancement for SARS-CoV-2. So it is not just about the presence of ADE, but actually it provides the circumstances or requirements of the ADE. When does the ADE occurs or occur? That's one. Secondly, they also have some suggestions to see how to prevent or minimize the chances of ADE. So that is the interesting part. They have done the experimentation in vitro, but they have used the antibodies from various monoclonal antibodies, plus they have used the serum from Moderna vaccinated individuals. So that is their antibody, and they have some control antibodies as well. So, beautiful study. What is scary in this is that yes, there is ADE possible. They demonstrated clearly, and I think it is repeatable, and there is ADE that occurs with monoclonal antibodies. There is ADE that occurs with vaccine generated antibodies. And there are times, phases when we get the antibody and when it starts waning. There are phases when there is a danger zone of ADE possibility. And so these are all the questions that if you can please keep in mind while we discuss this. So this is drbeen.com. If you like this work, there is a link in the description with a beautiful price. I always like to not call it cheap. It is really inexpensive and it is a one-time, non-repeating, non-recurring fee. So take advantage of that. Here is the study re-evaluation of antibody-dependent enhancement of infection in anti-SARS-CoV-2 therapeutic antibodies, the monoclonal antibodies, and mRNA vaccine anti-sera, that is the Moderna vaccinated individuals and their serums, using FCR, fraction constant of antibody receptor, and ACE2 positive cells. So that is the basic study. We'll discuss that. Let's do it. And I have seen some Twitter posts about this study where there is a lot of scare or concern demonstrated. However, for anyone to actually get the complete idea of what this study is saying, they should read it all the way down towards the conclusion as well, because there are some concerning aspects. I am concerned as well, but there are some solutions as well. Also, I would link my previous talks about ADE, and you would see that over the year and a half, my own discussions were presenting that we did not have evidence of ADE for SARS-CoV-2 at one point, and then we started having the evidence. This is, I think, the most clear evidence available with the correct circumstances. So these are our gifts for humanity. They are continuing. This is a Japanese study. I just showed you the study before. So the question is, why is this study done? So the authors said, we did the study because there are some previous studies, and they reference them. There are some previous studies that are done about ADE. However, those studies have inconsistent um, experimental setups. Some cells may not be the way they should be. Some expression of the proteins on the cells may not be exactly consistent across the experimentations. So even when ADE was shown or not shown, for example, they said that in one case, vaccine related experimentation demonstrated no ADE. However, the cell they used were actually not able to demonstrate the ADE in general. So they did this experiment, this study, to kind of have a consistent environment of when ADE occurs. And that actually gives an idea for the remaining newer experiments to use a similar requirements as well, or similar setups. So, for example, they say that in one of the studies, and so for here, FC receptor is a discussion that would occur very often. So, let me just very quickly explain what that means before we go further. So, if we have an antibody, so let's say this is an antibody. An antibody has a part of it that is called constant part, or fraction constant, or FC. And then it has another part that is called a variable part. And on the variable part, there is a binding region with which the antibody binds with various antigens binding region. So the discussion today is really about this fraction constant. Why? The binding region, this side, is used to connect with the antigen. For example, let's say here is a SARS-CoV-2, some scary looking dude, and that has this spike protein, and that spike protein is bound here. Then this antibody, now this is called an antigen antibody complex, and this antibody is tumbling around in the tissues and fluids and circulating in the blood as well, this complex. These complexes are then cleared out by the immune cells. So one such example is, for example, let's say macrophage, or a dendritic cell, or a neutrophil. Many cells do this. They have receptors on their surface that can bind with the constant region of the antibody. This receptor is called FC receptor, meaning receptor for the fraction constant of the antibody. The benefit of this binding is that this cell, let's say it's a macrophage or a dendritic cell, it is going to pick up this complex, internalize this, and destroy it. However, in the antibody dependent enhancement, this complex can enter the cell, because that is how this receptor works. It pulls the complex inwards, phagocytoses it, or endocytoses it. However, this virus can then escape that endocytic vacuole, come out of it, and cause infection. And now we have an ADE, antibody dependent enhancement. So FC and FCR, FC is this part of the antibody, and FCR is the receptor. So with this little segue, they say that in one of the experiments or studies, the researchers looked at the cells, or they used the cells that had only the FC receptor on them, but they did not have ACE2 on them, for example. In other studies, there were researchers who used cells that had ACE2 on them and not FC receptors on them, and so on. So they said because of this, the environment was different. So here, in one of the studies, FC receptor were present, plus ACE2 were present, and it showed that ADE was not observed with the monoclonal antibodies, and these were here. Similarly, there was another experiment, another study, where they had FC receptor positive and ACE2 positive, and ADE was observed. The point of this all is, they said these studies are inconsistent, so we wanted to have a consistent setup. So then, what was their setup? Their setup was that they used iPS immortalized cells. What are iPS? iPS are inducible pluripotent stem cells. What does that mean? A stem cell is a cell that can, when divide, it can make other daughter cells, and it can also make its own kind. That is the definition of a stem cell. Normally, in the experimental setups in the labs, we use various cell lines that are received from various embryos or human tissues of the aborted children. This is inducible pluripotent cell. Pluripotent mean a cell that can make many other kind, or a stem cell. Inducible means they took some skin cell or a blood cell from an adult, and then they converted it into a stem cell. This technology of this conversion, I believe it got a Nobel Prize some years ago. So now we can take an adult cell. You can, for example, donate your cell, and then they can induce that to go back towards a stem cell. They can open up the locked genes from it, and then they can use that as a stem cell. So that is the kind of thing they did. They had a stem cell formed, which were immortalized as well. So what they do is they take a cell and combine it with a cancer cell, and the cancer cells are immortalized. They have learned, the cancer cells, not to die. So if you take a normal cell and combine it with a cancer cell, now this cell becomes immortalized as well. So they took an iPS immortalized cell. This cell also has ACE2 on it, and TMPRSS2 on it, and FC receptor protein on it as well. So you can call these cells as stem cells, which are FC receptor positive and TMPRSS2 positive and ACE2 positive. And this is also something that I would like you to take away from this talk, that this setup is necessary. In our body, if a cell does not have FCR, that is FC receptor, plus ACE2 plus TMPRSS2, then that cell does not have ADE. Or conversely, any cell, for example, macrophages, dendritic cells, neutrophils, any cells that have FC receptor plus ACE2 plus the TMPRSS2, they can be a victim of this. Now, remember, all macrophages do not have ACE2, luckily, and dendritic cells do not have ACE2. That is how they can actually be protected. But still, this is the model of the cell which is the most prone or vulnerable to ADE. And they said this kind of a setup is highly susceptible to the infection. So that was their cell. Now, what was the experiment? What they did in this experiment was they took these cells that I just mentioned and they infected them with original variant. So they actually had the variants. They did not have the viruses. They actually had the viruses. I can only imagine how security protected this study was. So they had the original variant, they had the Delta variant, and they had the Omicron variant. They also had the antibodies from casirivimab. They also have mdevimab antibody. They also had sotrovimab antibodies. They also had serum from individuals who were vaccinated with Moderna, 1 dose, 2 dose, 20 days after the second dose, 52 days after the second dose, 90 days after the second dose, 175 days after the second dose. So they had a bunch of sera to understand how the antibodies were waning and how the ADE occurred as antibody concentrations changed. So this is the second takeaway that I can request you to hold in your mind, and that is the ADE was antibody concentration dependent. And you might be thinking this may be a higher concentration. Actually, no. A lower concentration of antibodies was the time when ADE occurred the most. So that is a very interesting thing. Lower. How low? I believe 1 nanogram per milliliter concentration of antibodies was the phase during which ADE was the most dominant. And there was also another problem. 1 nanogram per milliliter was so low an amount of antibody that it was not sufficient to neutralize the virus. So now, if the virus arrives in us at a time when antibodies are less, they are not sufficient, the antibodies, to neutralize the virus, but they're sufficient enough to bind with the virus and allow the virus another route of entry in the cell. Then that becomes the ADE vulnerability phase. So what did they see? I just mentioned it. So ADE occurs in a narrow range of antibody concentration. One more thing that is really important, the third thing I would request you to keep in mind, and that was this ADE occurred because of the antibodies that were neutralizing in their nature. So the more neutralizing antibodies present, and by more, I don't mean more in concentration, but the more aligned to neutralizing, the higher the propensity for ADE. So the ADE occurs in a narrow range of antibody concentration, and that was 1 nanogram per milliliter. It occurred in cells that were FCR positive and ACE2 positive. Now, the mechanisms to consider when virus is present and antibodies are present. There are two mechanisms that are opposing each other. One set of the mechanisms are protecting us. The other set of mechanisms are hurting us. So, for example, neutralization of the variant or virus by the antibodies. So here the setup is it's our body or the cells, FCR positive, ACE2 positive, one. Second, there are antibodies present. We're not talking about a setup where antibodies are not present. So we're talking about a person who was previously infected or vaccinated or had monoclonal antibodies. However, this particular study, did not talk about previously infected at all. It only talked about vaccinated or monoclonal antibodies. And you can ask this question that why? The reason is they were looking more into neutralizing antibodies behavior compared to binding antibody behavior. Cool. So now let's say neutralization by the antibody is possible when the virus arrives. So that is a protective mechanism. Then there is ADE, antibody dependent enhancement, which would of course occur only when the antibody is present. That is a negative. That actually increases the infection or infectiousness of the virus. And then there is cell mediated immunity, the cytotoxic T cells and the innate arm cells. The cell mediated immunity is protective as well. Then, if I can continue here, there are other mechanisms, for example, cytokine storm, which is hurtful as well, protective and hurtful. There is an ACE1, ACE2 imbalance, and that is protective and hurtful. So there are a bunch of mechanisms. And authors do mention in their manuscript that you have to consider the clinical empirical evidence in addition to these in vitro studies. Okay, so now the two opposing factors. On one end, we have cytokine storms, we have ADE that is hurting us in the presence of the virus and the antibodies. On the other end, we have antibodies to help neutralize the virus. We have cell-mediated immunity as well. So what they saw was, they said because of this mixture of effect, ADE increases the infection, neutralization, and cell-mediated mechanisms reduce infection. So then what did they do further? What they did was they had these cells, they call them in their manuscript, I believe, clone 35 cells. They had these cells, they cultured them, or they incubated them with casirivimab and imdivimab antibodies with a very low dose of the virus. This is an important point here. They were curious that is it the virus load that mattered for ADE, meaning more virus, more ADE, less virus, less ADE. So they, of course, started with a very tiny dose of the virus to figure it out that if the virus load was important. And they found out virus load had nothing to do with it. If the right concentration, the right bad concentration of antibodies were present, then even the small load of 40 copies per microliter of the virus were sufficient to cause ADE and allow the virus to enter the cells and become infect the cells and make the copies and come out and have more copies. So antibodies presence in a very tiny dose of virus is still sufficient to let the virus grow or expand. On the other hand, in the same experiment, they also had a control arm where they had other antibodies present, which were able to bind with the virus. However, they were not neutralizing type antibodies. So this control side actually did not have any viral expansion. So they put the cells, they put some control antibody in there. This antibody was actually able to bind to T cell receptor. Well, anyways, imagine useless antibodies. Some useless antibodies are present. They call them irrelevant antibodies. Same small amount of virus was present. If that small amount of virus was sufficient to cause infection, then in this control side, the virus would have infected. But they found out that on the control side, virus did not expand. But as soon as they had that with the antibodies, the same amount of virus, the same cells, the virus started growing in number, which meant it was the low dose of antibodies that were enabling the virus, and the virus dose did not matter. Now, they also did some more tests to kind of confirm their observation. So, what they did was they had the same thing done on a set of cells. That is, they had the virus plus the antibody low dose plus the cell, but the cell did not have ACE2 on it. Why? Because they wanted to reproduce the environment where there is just the FC receptor present in the cell, but no ACE2. And they wanted to see if that would work. And clearly, it didn't work, which is the strangest thing because the antibody binds with the FC receptor and then it can take the virus in through endocytosis. But somehow, ACE2 was necessary. At the same time, on the other cell, where ACE2 was present, the virus did not have the antibodies to bind with, and it didn't work. So it was clearly that ACE2 is needed. Whatever is that mechanism, they say actually, we are not sure why FC receptor plus ACE2 is necessary, but they both are important for enhancement of the disease. Then what they did was, and you might be thinking this as well, and you might say, maybe it was just the ACE2. It was the virus binding with the ACE2, and now we are just saying, hey, this is antibody dependent. So what they did was they blocked the FC receptor. So now the virus was only able to bind with the ACE2, but not FC receptor, and that also reduced the infection or blocked the infection, reduced the infection. So the environment had to be a cell with the FC receptor on it, with the ACE2 on it, with the antibodies present at 1 nanogram per milliliter, and then any of the variant. Interestingly, they also noticed that the current antibodies, monoclonals and the vaccine-generated antibodies, were able to help neutralize Wuhan variant, to some extent Delta, but to no extent the Omicron. And that is what concerned me, because in their manuscript they said the current vaccines against Omicron are only enabling the Omicron for, I'm paraphrasing them, are only enabling it to do ADE without neutralizing it. So the benefit of the vaccine for neutralization is gone or reduced. Actually, they actually say there is no benefit, or they don't say no benefit, they say no neutralization. However, ADE stays dominant. That is what concerned me. Okay, continuing. So as I just said, neutralization of the Omicron. They saw that with the casirivimab and imdivimab, there was no neutralization of the Omicron. We're talking about monoclonal for the time being. No neutralization, but ADE was occurring. So that means administering these antibodies to Omicron patients is actually counterproductive because the neutralization is not there, but ADE will occur. Then, this is the very important thing to keep in mind as a practical solution here. Sotrovimab was able to neutralize, plus it did not cause ADE. Sotrovimab was able to neutralize, plus it did not cause ADE. And so they speculated. They said the main difference of this antibody, other than the binding region, from the others is the following. The FC portion of the sotrovimab is changed. A couple of areas are changed in it for stabilization to make it a little longer living. And that change is in the FC portion. That change does not inhibit what is the name of this sotrovimab from binding with the FC receptor, but there is still something in it that the enhancement is not occurring. So they said maybe, and this is the solution they actually said in their manuscript. They said maybe antibodies for monoclonals or vaccine generated should have a change in the FC receptor, that it does not bind with the FC receptor avidly or at all. And now it only acts as a neutralization, but not as a binding. The problem will become that if it does not bind with the FC receptor, then how will you clear those complexes? So I think we'll have to take a solution like sotrovimab and make some modifications to the antibody so that it is still able to bind but does not cause ADE. I think that's a breakthrough discussion that they did. Then vaccines. So this is a very important part. So please, I need your attention here. They tested this vaccine with Wuhan, Delta, and Omicron. I'm only going to discuss Wuhan and Omicron. They saw that if they took serum of the people, individuals who got 1 or 2 doses of Moderna, what they observed was the following. After the first dose, 27 days after, there was no neutralization and there was no ADE. This is the first dose. So what they're saying is 27 days after the first dose, there was still no effect of the vaccine. Fine, that is understandable. Second dose, 20 days after and 52 days after, neutralization was there and some ADE was there, but neutralization was dominant. There were so many antibodies that they were able to neutralize, and so neutralization was dominant. So imagine this is that zone where there is some ADE occurring, but that is actually dominated by neutralization activity, and so the patient feels protected and is protected. However, day 98, this is their collection of blood, so day 98. I would say whenever the vaccine starts waning, day 98, second dose after, so day 98 after the second dose, there was no neutralization, but there was still clear presence of ADE. This is the danger part. So here, day 98, the antibodies have become less in quantity, which is a requirement for ADE. But when they become less in quantity or the titers are reduced, they're not able to neutralize very much. However, that is a perfect, the Goldilocks phase where the ADE is occurring. So this becomes the dangerous part. Now, can we predict that in terms of days? No, I think it is the prediction is based on the concentration, 1 nanogram per milliliter, so lower concentration. So this was Wuhan. Check out Omicron. That's even more serious. Why? I think you can already guess. Omicron is not able to be neutralized well. So 170 days after the second dose, there was no neutralization of Omicron by Moderna. They didn't use Pfizer, Moderna. However, they observed some ADE, and they use the word some ADE because I think they didn't see ADE in all cases. So maybe the 175 days was not the correct days. They may have to see a little earlier to have the right concentrations. But, anyways, they saw some ADE. And this is the bad part. As they increased the antibody levels from this 175 days, meaning as they increase the concentration of antibodies in the serum, the ADE started increasing as well. So it was a dose dependent ADE. And it required ACE2. So they concluded about Omicron, or they thought aloud with us, and they said, maybe Omicron is escaping in multiple ways. Number one, it is escaping the neutralization. And maybe it is also escaping by trying to find new ways. We know that it binds with CD147 more avidly as well. So maybe the ADE route is another route that it is preferring for infection. Now, the points I made so far were used by some of the folks to put on Twitter and kind of create a lot of concern. There are something positive in this manuscript as well. And I want to show you that. So first, let me show you those highlighted parts. So here, all of these green parts in their discussion are the ones that are actually positive. So I want to go over these as well so that we are not just sitting around saying, all right, we are effed. I think that there are, and you can also go to their discussion and see that. Number one, they said monoclonal antibody levels need to be lower to engage or create that danger zone of the ADE. So imagine this is that graph. Imagine this is the virus, and then we administer monoclonal antibodies. And I have made that into a straight one. So depending upon how many doses, it can go up. So let's say these are monoclonal antibodies administered. When these are administered, if they are functional, there are some that have just simply failed. But let's say sotrovimab, if they are functioning, if they're active against this variant, whichever variant the patient has, then the variant's quantity would start reducing. And also, in a few weeks, the monoclonal antibody would start reducing as well. Now, monoclonal antibodies, when administered, usually peak at about 192 microgram. It's not nanogram, microgram per milliliter. And then from there, they start reducing within 2 months or so and start going away. So they said that by the time the monoclonal antibodies titer, the level reduces sufficiently to bring us in that danger zone of ADE, by that time, maybe in 2 months, the virus has already gone from this person's body, ideally. And hence, the ADE is not a problem. However, my note here was, how about if somebody got reinfected here? How about if this was an HIV or immunosuppressed or compromised patient where the virus may hang in there for some more time? So there can still be a dangerous time, but the authors said that because this time of monoclonal antibody reducing in titer or concentration is so far out, couple of months later, the virus has to go out before that, and so we are okay. So that's one. And they said about the vaccine ADE. So imagine this once again with me as well. So here the green one, Let's say the vaccine doses are given, they produce the antibodies. We know that it starts 2 weeks after the second dose and then goes up and then wanes, right? So let's say this all is that curve. At some point, the concentration of antibodies will reduce and bring us in the danger zone where the ADE would start occurring. So if we become reinfected or infected here, we may be in trouble because of the ADE. And once again, ADE doesn't always mean that the person is going to get some severe bad outcomes. ADE would depend upon the person's health and what is the overall state of them. However, they said that it is possible that during this time of danger, the cytotoxic immune arm is still functioning. That is cytotoxic T cell, the CD8 positive cells, CTLs. They are functioning. They have not gone away. So when the virus comes in and we are actually in this dangerous time, maybe the cytotoxic cells will be protective. So that is their second point. These are those green little paragraphs that I showed you. And again, I have paraphrased them. Any mistake is mine, not theirs. Then the third thing, and this is something that I have been saying all along, and it almost seemed like they took that from some of my messaging, and that is because it's general scientific thinking. And that is, they said that volunteers who donated the plasma for their or the serum who were vaccinated, they said these volunteers themselves, after the vaccination 2 doses, stay infection free for 8 months. Then they got a third dose, the second dose. Then 8 months later, surely they reached a point of the danger zone, but they were okay. Then they got the third dose and the vaccine once again, the antibodies increased. So if empirically, if clinically the ADE was occurring, then they may have observed it in these donors, and they did not. So they said the same donor's plasma in the lab was showing ADE or was enabling ADE, but the same donors themselves were not showing that they caught the infection or had severe infection. So they said we need to rationalize that clinically in vivo the behavior of the antibodies may be different, or as I used to say, the overall body state, which is not just the antibody, but the cytotoxic cells, the innate arm, the nutritional status. There are so many cytokines, the mediators of inflammation, the liver's function, the function of the macrophages sitting in the spleen, the follicular dendritic cells. All of those things are working together to help us. But in the in vitro, we are only looking at a cell and an antibody and a virus. That has no C helper cells around, that has no mediators around, that has nothing else around. So that environment and our body's environment is different. So they said that in vivo outcome is different from in vitro, and we should keep that in mind. And then they say we do not know why it was different. And they said larger studies need to be performed to understand that a person's serum in the lab is enabling ADE, but the person is still protected. What is the reason? Maybe the person is just not exposed to the infection. Maybe the infection, when it occurred, would have caused ADE. So then they give a suggestion and they say they feel that the FC part of the antibodies should be modified so that the ADE's chances are reduced. So that is the discussion. I hope it makes sense, and I hope that it didn't carry that scare that I saw going around by some of the scientists and journalists about this study. This study has concerns. I told you when I was reading it, there are certain parts where I became concerned. At the same time, there are some interesting, positive parts of this all as well. So this is the discussion. Thank you very much for being here. Please do me a favor, like, subscribe, and share. or just like. That is the smallest fee that I can request you. And if you subscribe and share, that will be amazing as well. There are some links in the description. I would love it if you can join me on Patreon. It's $5 a month. And if you join it, that would be a great way to support this. You can also support by PayPal or Substack or becoming part of YouTube, or you can buy Dr. Bean's plan. The link is also in the description. So let's see if there is any comment. Very good. So, all good. Thank you very much. Have a good night. Please like, subscribe, and share. And I would see you tomorrow.